Good evening everyone from New Zealand, and good morning to you. Extremely sorry that I'm not with you in person and enjoying all that Rome has to offer. For those of you who don't know me, my name is Greg Macpherson. I'm a pharmacist and I made the jump to biotech over a decade ago, initially focusing on the impact of mitochondrial dysfunction on longevity before turning my attention towards a more holistic view of the aging process. Most of you will be aware of the Hallmarks of Aging by López-Otín, published in 2013. This paper is incredibly interesting, as whilst it described the key drivers of aging, it also created a framework for targeting aging, and that's been the focus for me over the last 5 or so years. The work culminated in me writing a book called Harnessing the Hallmarks of Aging, and that really helped me crystallise my thinking on these processes and how they all interconnect. What you're going to take away from this talk is that the hallmarks aren't just interesting theoretical concepts. They're genuinely targetable and measurable, which means that we can actually intervene on them and track whether these interventions are working. That's what makes this framework so powerful. The aging challenge. That's why we're all in the room or dialing in. It is incredibly apparent that aging is the single biggest risk factor for disease. The older we get, both chronologically and biologically, the more chance we're going to get upfront and personal with the disease that's associated with aging. Reversing and slowing the aging process has to be the single biggest priority for individuals, but also the countries that we live in, as populations continue to age and fertility rates continue to drop. To put up some numbers around this, by 2050, 1 in 6 people globally will be over the age of 65. That's up from 1 in 11 in 2019. Japan is already living with this reality. Over 29% of their population is over 65, and they're experiencing this firsthand. The economic impact is staggering. Healthcare costs for elderly are 3 to 5 times higher than for younger adults. The dependency ratio is already putting strain on economies. We're witnessing healthcare spending accelerate, and at the same time labour markets are contracting, and there will be increasing intergenerational inequity. This issue is the perfect example of a slow emergency. Unlike pandemics that grab headlines and galvanise immediate action, aging just doesn't make the news. But here's the thing. Aging is the single largest contributor to global disease burden. We just don't see it that way because it's happening so gradually. We need to be incredibly focused on solving aging if the next 20 years is going to be sustainable. Luckily, we're arriving at a point in human history where we're starting to solve the problem. We've got new tools, we've got new understanding, and I am very optimistic. The hallmarks of aging were identified as a framework to understand the aging process. Think about the germ theory for a moment. Before we understood that pathogens and microbes caused infections, doctors were treating symptoms without understanding the cause. They didn't know what they were fighting. But once we identified the actual culprits, we were able to develop antibiotics and save millions of lives. That's the paradigm shift we're seeing with the hallmarks. They give us clarity for aging. Now that we understand the underlying causes, the hallmarks give us a framework to modulate aging, and it is just a matter of time before the first antibiotics for aging are validated in the clinic and show benefit in population health. This framework also allows us to shift the language we use. Instead of talking about anti-aging, which sounds like cosmetics and skin cream, we can talk about longevity medicine. That sounds like science, and it sounds legitimate. Now, I have to say the skeptic in me is concerned about one thing, and that is that until we develop interventions that get around lifestyle issues, we continue to float down the proverbial creek without a paddle. We've been unable to successfully drive population-wide behavioural change globally. We know what works: healthy eating, exercise as a cornerstone of culture. But look at the blue zones. Even there, as Western diets and sedentary lifestyles infiltrate, we're seeing health outcomes decline. These are foundational elements that pockets of the world are getting right, but they remain the the minority, and we really need to focus on strategies at a population level to start to get that back on track. Now, I'm not going to go through each of these hallmarks in detail because they'll be familiar to you all, but I do find it very interesting that 3 of the 4 primary drivers of aging are directly linked to DNA. It's very clear that we're looking after your DNA and the machinery that enables it to operate effectively, repair itself and maintain itself is absolutely critical. And here's something that really fascinates me. The 9 hallmarks are organised in a hierarchy: primary, antagonistic and integrative. Understanding this structure is crucial. The primary hallmarks cause the initial damage, things like genomic instability. That damage then triggers antagonistic hallmarks, which are basically the body's compensatory responses. The problem is these compensatory mechanisms help in the short term but become harmful over time. Take cellular senescence as an example. Cells stop dividing to prevent cancer, which is protective, but these senescent cells accumulate and cause inflammation and tissue destruction. Then we get the integrative hallmarks, the final functional decline, like stem cell exhaustion, where we just can't replace damaged tissue anymore. Understanding this cascade is absolutely key to knowing where to intervene and what is going to have the most impact. We also have epigenetic clocks embedded in our DNA, which give us a read on how old our cells are actually behaving. The work that David Sinclair and other teams are doing regarding reprogramming these clocks is incredibly interesting and ultimately likely to be the pathway to significant lifespan extension. In 2023, López-Otín and colleagues updated their framework and added 3 more hallmarks to the roster. Let me tell you about them. First, we have macroautophagy. Essentially, that's the cell's recycling system. When macroautophagy works well, damaged organelles and proteins are cleared away. When it fails, damaged components accumulate like rubbish piling up in your house, and that's a real problem for cellular health. Second, chronic inflammation. This is probably the most interconnected hallmark. It touches every other one. We're talking about low-grade chronic systemic inflammation, and it's now recognised as a key driver across the aging process. Third, dysbiosis, changes in our microbiome. There's the gut-brain axis to consider. Your microbiome affects everything from cognitive function to immune regulation. When dysbiosis occurs, these systems start to decline. The expansion from 9 to 12 hallmarks shows the field is maturing and getting more granular in its understanding of the aging process, and I'm sure this list will expand further and possibly contract again as we get better at modulating the primary drivers and discover which are the true predominant targets for interventions. Once we resolve the upstream issues, then the downstream hallmarks should fall back into line. What is a hallmark? It's very simply defined. First, they get observably worse during aging across mammalian species. Second, if we accelerate them in the lab, we see accelerated aging. And third, if we stage an intervention, we see reversibility. That third point, therapeutic reversibility, is what separates hallmarks from mere biomarkers. If we couldn't reverse them, they'd just be interesting observations. But the reversibility criterion means that we have actual targets for intervention, and this is what makes this framework so powerful. Consider senescent cells. We know that clearing senescent cells in mice extends both health span and lifespan. That's the reversibility criterion in action. We're not just observing something, we're actually intervening and seeing real improvements in aging. This framework essentially gives us a checklist to evaluate any proposed aging intervention. Does it hit one of the hallmarks? Can we reverse it? If not, it's probably not going to be as impactful as we hope. The next group of hallmarks are emerging candidates that the field is seriously considering. I can see 3 hallmarks that relate to the structural and mechanical changes occurring within the extracellular matrix. This is the scaffolding that surround our cells and organs. These came out of a meeting in Copenhagen in 2022 where leading aging researchers got together to evaluate candidate hallmarks. One of the most interesting is the extracellular matrix deterioration. Why? Well, it's because something that people experience directly. You feel it as skin aging, you feel it as joint stiffness, and you feel it as arterial stiffening. These aren't abstract molecular changes. They're things that people actually notice in their bodies as they age, and that's what makes the ECM such a compelling hallmark. There's also a fourth hallmark associated with DNA, the loss of fidelity linked to gene expression patterns as we age. That's a nod to the informational theory of aging, although my sense is that epigenetic reprogramming will likely resolve this issue. And since the ECM deterioration is so important and interconnected, that's what we're going to dive into in the next slide. The extracellular matrix as a hallmark of aging is an area of real interest. The ECM is essentially the scaffolding of our bodies. It provides the structural support, it creates the signaling environment the cells depend on. When it breaks down, cells lose that support and those crucial signals, and everything downstream starts to fail. To put this in perspective, collagen makes up about 30% of the total body protein. So we're not talking about some minor component here, we're talking about a massive structural element. The work of Colin Ewald in this area is fascinating, and I really encourage you to read his work. It'll change the way that you think about aging. And now here's something really interesting about diet. Advanced glycation end products, or AGEs, from diet actually accelerate ECM damage. Cross-linked sugars bind to collagen and damage the structural integrity of the matrix. So there's another connection back to the lifestyle point that I made earlier. What you eat directly impacts ECM health. Whether we can reverse this damage once it's occurred remains an open question, and it's why we should all be living a longevity lifestyle right now while we wait for interventions to make their way through to the clinic. And here's something most people don't realise. All those folk knocking back collagen supplements to keep them looking beautiful at skin deep level are likely to be getting more systemic benefit than they think. The global collagen market is valued at over $9 billion, and it's driven largely by beauty consumers. But there's great evidence that collagen supplementation may actually be helping them slow aging systemically, not just cosmetically. The beauty seekers might be inadvertently doing the right thing for longevity. So let's turn to targeting the hallmarks of aging. To get a top-level view of the progress that's being made, what's really exciting about this moment is how quickly the field has shifted. Just over a decade ago, we were really in observation mode, trying to understand aging. Now we've moved to intervention mode, and we're seeing that both pharmaceutical pathways and nutraceutical pathways are being explored simultaneously. That's a really significant acceleration. And the convergence of evidence suggests that we're getting very close to real therapeutic options that could meaningfully extend health span. Senolytic therapy is well established as a way to lower the inflammatory burden that senescent cells increasingly put on the body with age. Companies like Unity Biotechnology and others are running human trials right now. The dastanab and quercetin combination that's been used in many studies was actually pioneered by James Kirkland at the Mayo Clinic, and it's shown real promise in animal models and early human work. We're at an exciting point where senolytics are transitioning from research curiosity to potential therapeutic reality. mTOR inhibition via rapamycin and its analogues is a remarkable intervention that triggers cellular pathways associated with conserving life and make perfect sense as an organism switches into longevity mode in the absence of nutrition to just give it more time to find life-sustaining food. The Pearl trial, that's the participatory evaluation of aging with rapamycin for longevity, is generating real-world data that's going to help us understand whether these effects translate to humans beyond the laboratory. It's a landmark study, and the results should inform whether rapamycin analogues might become part of a broader anti-aging strategy. NAD precursors have had a huge amount of research behind them, and we should continue to explore other compounds that decline as we age. Now there's actually interesting debate in the field about bioavailability, whether NMN or NR is more effective, but what's emerging is that the combination approaches seem to work better than simple agents. The work that Rajesh Singh has done around taurine and Dudley Lamming around calcium alpha-ketoglutarate is aligned with this broader strategy of targeting metabolic factors that decay with age. The field is converging on the idea that you need to hit multiple targets simultaneously rather than betting on a single molecule. I'm sure that there will be plenty more compounds to be surfaced as research continues and as we understand some of these changes that occur as we age. And one of the most important in my mind is the epigenetic reprogramming pathway. I think this is our strongest lead. It targets the primary hallmarks and is the intervention that is furthest upstream. The investment in pouring into this space is extraordinary. We're talking billions of dollars going into companies like Altos Labs and others working on partial reprogramming and epigenetic reset technologies. The science is compelling. The financial backing suggests serious investors believe that we're on the verge of real breakthroughs. This is not fringe research anymore, it is mainstream. There was some fascinating work done years ago that compared the biological strategies of senescent versus non-senescent animals. It turned out that senescent animals had an overarching survival benefit because they lived short lives and reproduced rapidly, enabling them to adapt quickly to environmental changes. Those senescent animals are our distant forebears, and we've inherited their biology. This is actually key to understanding why cancer and aging are so intertwined. Senescence itself was an evolutionary strategy to prevent cancer by limiting cell division and triggering cell death. The same mechanisms that protected us from cancer are literally driving the aging process. It's perfect for getting us to reproductive age in harsh ancestral environments, but perhaps it's time to engine ourselves out of that very self-limiting pattern and borrow strategies from non-senescent organisms like reptiles that just don't age the way that we do. I got the chance to speak to João Pedro Magalhães a year or so ago, and I asked him what was the one discovery in recent times that's been the most interesting. And he said biological clocks without hesitation. Steve Horvath's original 2013 epigenetic clock was truly a watershed moment. For the first time in history, we could measure biological age objectively. Before that, we had no way of quantifying aging at a molecular level. That we all have epigenetic clocks ticking away on our DNA that reflect our biological age is absolutely remarkable, and it's one of the strongest clues that aging is programmed. Researchers like David Sinclair and his teams are working on interventions to establish whether we can reprogram these epigenetic markers and in effect reprogram aging. And I think the answer will ultimately lie in understanding those cells that don't appear to age. Clinical trials over the next few years will be very revealing and tell us if this is going to be an effective approach. Like all nascent technology, each generation of biological clock is getting better, and we're now getting insight into organ system aging. The granularity of this is absolutely stunning. We can now see that someone might have a 50-year-old heart, but a 65-year-old liver. That kind of resolution changes how we think about prevention, and it's going to usher in an era of true preventative medicine as we identify which organ is going to be our individual limiting factor. I've got Crohn's disease, and it's incredibly interesting to me whether I would have been able to identify this early and modify the disease process if I'd had that kind of organ level testing. And I suspect there's someone in the room dealing with a chronic condition. Maybe it's your gut, your brain, your heart. This is personal for all of us. We all have a weak link somewhere, and the promise of organ level biological age testing is identifying that weak link before disease manifests. What's exciting is these tests are now commercially available through companies like True Diagnostic. The cost has dropped dramatically. We're talking from thousands of dollars down to a few $100s. The accessibility is game-changing because more and more people now can track their biological age trajectory and make targeted interventions. Personalised medicine is finally getting ahead of disease rather than chasing it, and that's just incredibly exciting. So into the clinical setting. 5 years ago I founded SRW Laboratories to build a natural ingredient protocol to slow aging, as I believed if we took a 360-degree approach across the hallmarks of aging, we would deliver health spin benefits. Immediately after launch, we got signals back from our customers that our protocol was working, but we needed to do the work to better understand whether we had something interesting or not. 3 years ago, we made the decision to invest in a clinical trial. For a startup, that is a serious commitment. It requires capital, expertise, regulatory navigation, the whole lot. But we realised that without evidence, we'd just be another supplement company making claims. We needed to know that we actually had something meaningful. So we designed our trial to validate our theory that targeting the hallmarks of aging would modulate the aging process. We also partnered with Varun Dwarka and the team at True Diagnostic, who were instrumental in the epigenetic analysis. That collaboration was crucial. They brought the expertise and the validated testing program that made our data credible. With the usual budgetary constraints that afflict startups, we ventured forth. We developed 3 products that were strategically designed to tackle different hallmarks of aging. Product 1 targeted the primary hallmarks. Product 2 focused on energy and metabolic hallmarks, mitochondrial dysfunction, and addressing dysbiosis. And product 3 targeted cellular housekeeping, the clearance and maintenance hallmarks, senescence, proteostasis, and autophagy. By the time you hit 50, you should be addressing all of these in parallel. We worked with a group of adults in the States and asked them to take our protocol daily alongside walking for 10 minutes and 5 minutes of meditation. Now we included those lifestyle components because the evidence for their impact on biological age is solid. Walking and mindfulness aren't just flashy, they work and they represent accessible interventions that anybody can do. We measured both biological and physiological age markers at baseline, 3, 6 and 12 months. We deliberately chose a mean age of just over 65 years, old enough to show measurable aging markers, but young enough to show potential benefit from intervention. That's the sweet spot for seeing real change. The result was remarkable. A wide range of biological age clock reversal, with our top score close to 5 and 3/4 years of age reversal. And if you add back the half year that the adjacent population moved forward, we got closer to a 6-year benefit using the system's age clock. For context, Greg Fay's TRIM trial was considered groundbreaking, showed 2.5 years of reversal. We're talking about nearly 6 years. The statistical significance on both system age and PC grim age gives us confidence that these aren't just random fluctuations. There is actually something real happening here. This was a single-arm study with each participant being their own control, a limitation I will cover in a moment. But I want to note that while omic age result didn't reach statistical significance, the direction of effect was consistent with the other clocks, which strengthens the overall narrative. We retrospectively looked at an identical population to capture the delta between those in the study and those not, but in the next trial we will include a placebo group so we capture true head-to-head comparisons. A very interesting aspect of this trial was that the impact of the intervention across the 4 time points measured. You can see that the different clocks showed different temporal patterns. Some measured immediate improvements that declined over time, some reflected initial acceleration of aging before changing direction, and particularly interesting the Dunedin pace clock, which measures pace of aging rather than absolute age. The fact that it slightly accelerated was interesting, and perhaps reflects that the cohort that is driving the algorithms are in their 50s now, and our cohort range from 50s to mid-80s. The damage clock is interesting too. It showed the most dramatic early improvement, dropping from 2.46 down to negative 2.07 at the 3-month mark. That kind of quick, rapid shift suggests that we're seeing quick clearance of damaged cells. The different temporal patterns across these looks likely reflect different biological processes being captured. One might be about tracking cellular stress, the other mitochondrial dysfunction, the other epigenetics drift. This complexity is why single time point studies can be very misleading. You need to see the full arc to understand what is happening. And while epigenetic clock data was really encouraging, there's still a gap as to what biological age reversal actually means. Are they recording verifiable modulation of the aging process? And what does that ultimately mean for health span and longevity extension? That data is going to take some time to translate, but from my perspective, these clocks are allowing us to identify therapeutic aging interventions, and it is just a matter of time before that becomes unequivocal. As part of our study, we measured physiological markers of aging, grip strength, chairs, stand test, timed up and go, etc. These are standard measures that have been used in the clinic and are directly linked to longevity. It was incredibly reassuring to see that these physiological markers of aging aligned with what we saw with the biological markers. Our study participants came out of the year trial stronger, leaner and lighter. Not Ozempic lighter, but the weight loss was modest and accompanied by weight circumference reduction, suggesting loss of visceral fat specifically, which is the metabolically most dangerous type. The specificity matters because it argues against pure placebo effect. If this was just an expectation or motivation, we'd see uniform changes. Instead, we saw very targeted improvements. The improvements to grip strength are meaningful. Grip strength is one of the most strongest predictors of all-cause mortality in older adults. A 3-pound improvement might sound small, but it's clinically relevant. And the chair stand test improvement was equally important for falls prevention, which is a major cause of morbidity and mortality in older populations. Curiously, stem cell division rates were significantly reduced at the 3-month mark, which is very statistically significant. This was measured using the EPITOC2 clock, which is a mitotic clock that counts the cumulative number of stem cell divisions. Higher numbers correlate with both aging and cancer risk. Reduced stem cell division is good news because every time a stem cell divides, there is a chance of a copying error. Fewer divisions means fewer opportunities for mutations. The rapid initial effect suggests the intervention quickly reduced cellular stress, allowing stem cells to return to a more quiescent state rather than being in constant replication mode. Now, it might seem a leap to suggest all of this means for overall cancer risk, but given the mutation risk with each stem cell division cycle, we think the intervention lowered overall mutation risk. This is an integrative finding. Stem cell exhaustion is one of the hallmarks of aging, so seeing improvement here suggests that we're getting upstream benefits that cascade through multiple biological systems. This needs to be explored further in larger studies, but it's a very promising signal. Beyond the epigenetic clocks, we saw immune cell population remodeling, reflecting a younger phenotype, and other data is also associated with gene expression, reflecting a younger animal, so to speak. Now let me dig into the immune data more specifically. We saw a significant increase in naive CD8 T cells. This is really important to understand. As we age, our immune system becomes dominated by memory cells, cells that have been activated in response to previous infections or antigens. The problem is that we lose the ability to generate new immune responses to novel threats. So an increase in naive CD8 T cells suggests that intervention is actually rejuvenating the immune system, restoring the ability to mount fresh responses. At the same time, we saw decreases in CD4 memory cells and Treg cells, which suggests reduced chronic immune activation. That's a good sign. It means the immune system is less chronically activated and less inflamed. On the DNA methylation side, we identified over 1,600 differentially methylated CpG sites. That's a substantial number, and it tells us that the intervention is having a broad epigenetic effect across the genome. When we looked at genes and pathways that were being hypermethylated, we found ones that protect against oxidative stress-induced senescence, and pathways being hypermethylated, involving collagen biosynthesis and tissue regeneration. This is exactly what you'd hope for in an anti-aging intervention trial. And this methylation data gives us molecular mechanistic evidence of why our epigenetic clocks are improving. One of the key genes we identified was Has3, which codes for hyaluronic acid synthase 3. That's directly relevant to tissue hydration and ECM maintenance. This is exactly the kind of molecular detail that helps us understand not just what something is working, but it's why it's working and what biological processes are being reactivated. A key finding was that in general, across all the epigenetic clocks, we noticed that we made things worse early in the trial, and then the magic started happening in the second half of the trial. This is suggestive of an impact on senescent cell population remodeling, which is consistent with the senolytic stress response. It also shows the importance of longer-term intervention trials, because if we'd stopped that trial at 6 months, we would have been beyond disappointing. The challenge now is to understand whether we extend our trial length on our next program, or whether our protocol is better with cycling. Think about it like starting a new exercise program. When you start, you feel worse before you feel better. Your muscles are sore, you're tired, your body's stressed, but that stress response is exactly what triggers adaptation and improvement. The same principle appears to be at work here at the molecular level. The initial worsening is what we see in the first half of the trial. It's likely the body's stress response to the intervention, and the improvements in the second half are the adaptive response. On the cycling question, whether periodic intervention followed by recovery protocols might be more effective than continuous dosing, we don't know. There is emerging research on pulse dosing protocols for neuroleptics, and that may be the way forward. But what we really need to do is to continue that research and look at cycling, or whether we actually see continued benefit by continuing with the trial. So what were the key learnings from the study? Well, I want to pull this together, the threads of what the data is telling us and what it might mean for the future of longevity medicine. The results we've shown to you are really encouraging, but we also need to maintain scientific rigour as we move forward. That means bigger studies, more controls, and better mechanistic understanding. And that's where we're heading next. We believe we've validated the theory of that holistically targeting the hallmarks of ageing is a reasonable platform as an intervention to modulate ageing. One molecule, at least in the nutraceutical space, is unlikely to deliver the benefits of a multi-target strategy. So for now, compound formulas that target pathways associated with ageing are going to deliver results and are the next stage of work that we need to do to continue to explore the cellular pathways involved with the hallmarks of ageing and get better at targeting them. And let me emphasize what this table really represents. It's the core thesis of everything we're doing, that each hallmark can be targeted with specific interventions, and this trial provides measurable evidence for each one. But the interconnected nature of the hallmarks means that when you improve one, you will get cascade benefits in others. For example, we improved mitochondrial function via NMN, but we also reduced oxidative stress, which again helps with the genomic stability. Everything is connected. Now, the pharmaceutical industry has been very slow to adopt multi-target approaches because single molecule drugs are much easier to patent and regulate. But the evidence is increasingly suggesting that ageing requires a systems level approach. Nutraceutical formulations have the advantage of being able to work and combine multiple actives that work synergistically. The next phase of our research is going to focus specifically on understanding which ingredient combinations are driving the most significant effects. There were limitations to our study. We did not have a placebo control group. Our sample size gave signals, but we need larger powered studies to validate. We asked people to walk and meditate. We don't think it did, but perhaps it did have an impact on the outcome. So our next study needs to deconvolute lifestyle elements from the intervention. We need to add in more data points to really get amongst the weeds. And lastly, we need to look at groups that cycle the protocol and groups that go beyond 1 year. I really need to be candid about this. We are aware of the limitations, and they are significant. We don't shy away from it. The scientific community rightly demands rigour, and that's exactly what the next trial will deliver. We will plan to raise the bar on this. And when we talk about adding more omic data points, we're really going to integrate proteomics and metabolomics aside genomics. This is going to give us a much richer picture, and right now we're looking at what's changing at the DNA level. With proteomics, we'll see what proteins are going to be produced in response to that intervention, and with metabolomics, we'll see what the metabolic pathways are shifting. This integrative approach is going to give us a much deeper understanding of the mechanism of action. We're also actively exploring partnerships with larger institutions to increase the sample size and geographic diversity. The work that began in our lab needs to scale up, and we're having conversations with groups that got that infrastructure to make that happen. The key takeaways I'd like to leave you with: the hallmarks of ageing framework is targetable. What I mean about that is that the hallmarks give us a common language and framework which unites researchers across disciplines. Whether you're a cell biologist studying senescence or a clinician treating patients, the hallmarks give you a shared vocabulary that's incredibly powerful. Multi-targeted strategies show promise, and while pharmaceutical companies are pursuing single molecule blockbusters, the evidence suggests increasingly that ageing requires a systems level approach. You can't solve a complex problem with ageing with a single bullet. Biological ageing is modifiable, that is critical. 10 years ago we couldn't really measure it reliably. Now we have valid, reproducible tools, and we can measure it accurately and show that it does respond to intervention. That changes everything, and it means we've got a target to shoot for. And finally, we need to keep unpicking the mechanisms and protocols as we work towards improving and formulating products with significant evidence of effect. This kind of work is going to enable us to accelerate progress across the entire field. Studies like this will help progress the translation of research towards the clinic. The long march continues, and ultimately we will find the perfect combination of therapies. And picture this, within the next decade, getting your biological age tested could be as routine as getting a blood test at your annual physical. I believe this is the future we're moving towards, and just as cancer treatment has moved from one-size-fits-all chemotherapy to targeted therapies based on specific genetic profile of your tumour, longevity medicine is proving towards personalised protocols based on your individual hallmark profile. Maybe you've got a particularly strong mitochondrial dysfunction, so we weight the intervention towards NAD boosters. Or maybe your hallmark issue is senescent cells, so we focus on senescent. That kind of precision medicine is coming. The gap between longevity science and clinical practice is closing fast. New diagnostic technologies from partners like True Diagnostic will enable this, and conferences like the one we're here at right now is crucial for building the bridges between researchers, clinicians, and the public. This is where translation happens, and we are at an inflection point where what used to be science fiction is genuinely becoming science fact, and that is incredibly exciting. I want to thank the conference organisers for putting together such an incredible event, and all of you for giving me your time and attention. I also want to acknowledge my research team and our collaborators, particularly Varun Dwarka and the team at True Diagnostic, who have been instrumental in helping us measure biological age in such rigorous ways. And of course, this work wouldn't be possible without our study participants who took the time to be part of this investigation. That kind of commitment to advancing science is so invaluable. Again, I want to thank you. I'm happy to take questions, but you'll need to fire them through to my email that you've got below. And I wish you well for the rest of the conference. Thank you very much.